This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 483, recorded on March 2nd, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here inside a warm, comfortable house. Because outside it sucks.
Pretty bad, yeah?
Yeah, I got this northeaster blowing through. It's not as bad as Boston is getting right now, but winds 22, gusting 38, and rain, overcast, layers and layers of clouds, and just really wet, cold, yuck.
We have 3 degrees Celsius, mixed rain and snow here in New York with 28 mile per hour winds, low visibility. Yeah. It's not a great day. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. So today, blue sky, white puffy Springfield clouds. Yesterday, however, they said we were going to have rain, and for southeast Michigan, it was going to change to a dusting of 1 to 2 inches of snow. So right away, it's a little suspicious because 2 inches is never a dusting. And then it turned into 7 or more inches of heavy, wet snow.
Yuck.
It was really horrible. So.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
We don't want to hear about your weather.
You're going to have to hear it anyway. We got 67 degrees. That's 19 C. And I would say partly sunny. You know, there's more clouds than I like, but I can handle it. There's some sunshine here.
Are you wearing shorts, dude?
Not right now. I am wearing a short sleeve T-shirt, and I got I'm in my bare feet. But this is my home office here, so I can do what I want. We got 62% humidity. That's a dew point of 53 degrees. All in all, it's pleasant, and we got springtime going on here. Things are turning green. The red buds are turning red. So it's all good.
All right. I'm glad one of us has good weather.
Yeah. Well, it sounds like Kathy has pretty good weather comparatively.
Yeah, now.
That's true.
Yeah.
Yeah.
Is there still snow on the ground, Kathy?
Oh, yeah.
Where can science, technology, engineering, and mathematics, STEM, take you this April? Discover how STEM can take you under the microscope and beyond with the American Society for Microbiology. ASM will be at the USA Science and Engineering Festival on April 7th and 8th at the Washington, D.C. Convention Center. Stop by booth 6200 to explore microbiology with infectious hands-on activities. Hope they're not too infectious. Yeah. Hope that maybe they have a Gesundheit too. Registration is required for this free event, so visit usasciencefestival.org to register. This week we did infection basics in virology, and I told my class about the Gesundheit too. They were laughing like crazy.
Yeah, that's great.
They loved it. So go listen to TWiV and hear all about it. I wanted to relay an interesting experience I had. I bought a HomePod, which is this Apple speaker thing that you talk to.
So you're one of those people who deliberately bugged his house. This should be good.
Yeah. That's the same thing my cybersecurity son said. Dad, you're going to get hacked.
Yeah.
Anyway, first thing, what do you think is the first thing I ask Siri?
What's the weather?
No.
Oh.
What is a virus? And my wife rolls her eyes, of course, but unfortunately, Siri said she read from the Wikipedia entry for computer viruses. Yeah. I guess if you
no, the kind that make you sick.
And so I'm sad because if you search Google for what is a virus, you get a definition. And the first definition is an infective agent. And the second is a piece of code. So I suppose a little computer hardware-y speaker would pick the computer virus first. But I wonder if anyone has a Alexa or an Echo or a Google speaker thingy, try asking them what is a virus and see what they answer. I'm wondering if one of them gets it right.
I think you also ought to ask them if viruses are alive.
That would be good.
We haven't done that for a long time.
Then the second
They'll probably
question,
just say, "This is what I found on the internet."
Yes.
Or blah, blah, blah.
So the second question I asked was, who is Vincent Racaniello?
Did it reach your Wikipedia page?
Your wife rolled her eyes again.
She rolled her eyes even more. And then my daughter said, "Stop it, enough." And yeah, Siri read the Wikipedia page. She started to read, and I said, "Okay, it's enough. Stop." And the funny thing is, you say, "Okay, stop." She'll go, "Uh-huh." Or, "Hey, Siri, uh-huh." It's funny. Anyway, I just like to have it play music mostly. I think if you're doing something in the kitchen and your hands are wet, you go, "Hey, play some music." Kind of cool. All right, follow up. Chaim writes, "Dear twivmasters, in episode 482, the paper discussed used the PacBio long-read sequencing technology. However, Vincent referred to the machine as being the size of a harmonica, when the PacBio sequel is actually 5.5 feet tall. The Minion by Oxford Nanopore is the small portable long-read sequencer that had been mentioned on TWiV. That's right. And I heard long-read, and I just put two and two together.
Could be a large harmonica.
Well,
Yes.
why would they have to build a 5.5 foot tall I really 5.5 feet teeth? That's ridiculous, isn't it?
I think it may operate a little faster, more efficiently, more
I'm sure.
repeatedly.
and with better fidelity so far.
Yeah.
But really, if a refrigerator, that's I'm sure it's going to get smaller.
Well,
But
how much smaller does it have to be?
I don't have any room in my lab for a 5.5 feet tall thing, but I guess I wouldn't get one. How much smaller does it have to be? Yeah, I suppose. But I love the ones you put on your desktop, right?
Oh, that's a very cool concept, and that you could take that to places that you couldn't take anything larger. But I don't know, in the larger form, they can pack in, so to speak, a lot more stuff.
Well, my apologies to the makers of Minion.
Minion.
But I like
Min.
I know.
But the little guys who run around with yellow.
Yes.
Right? They're so
Yeah.
cute. Rob has some interesting information. Dear Twivers, a quick error correction email on your discussion on PacBio sequencing from the last TWiV, which mixed up two different systems. There are two commercial long-read sequencing technologies, PacBio and Oxford Nanopore. PacBio systems covered last week are the size of a U.S.-style refrigerator, not a harmonica, as Vincent suggested. Let me just pause here. Are our refrigerators bigger than other countries? They are?
Yes.
Yeah.
Well, they're bigger than UK refrigerators by a factor of about 2.
Wow.
At least 2.
I didn't realize that. All right.
Well,
At least
or
that was
now.
the case.
Yeah.
At least that was the case 40 years ago when
That's funny.
I experienced this.
They turn linear double-stranded DNA into what is topologically a single-stranded DNA circle by ligating a hairpin oligo to each end. That's cool. They then sequence round and round the circle, covering forward and reverse strands, sometimes more than once each to reduce error.
That's cool.
There you
I
go.
like that.
But I don't know why you need a refrigerator to do that. Why couldn't it be smaller? Oxford Nanopore systems, the Minions, as Kathy would say, are the size of a harmonica and reads the nucleotide sequence of linear DNA that is fed through a pore. And this is Rob in London, where it's -2 Celsius, snowing pretty heavily, and everyone is hiding indoors because the UK can't cope with snow. And he sends a YouTube video about that, which is not safe for work.
But it's hilarious.
Well, if you wore headphones, you could watch it at work. There's nothing bad in the images in it.
That's just cursing, right?
Yes.
Yeah.
Rob is at Imperial College London, which, if I remember, Dr. Condit used to be there.
No, I was at the Imperial Cancer Research Fund.
Oh, I got the two first letters right.
You got the first, yeah, that's right.
I'm going to have a goal of every week making some mistake. Now, about this, the other thing I wanted to say. Oh, so Rob says topologically, right? So I have a study question for my virology class. Be familiar with the different topologies of DNA and how they are replicated, right? And 10 students come to me and go, I don't know what topology means. I said, well, the landscape and all that. Yeah, I know that, but when you're talking about DNA, I don't know what it means. Isn't that funny? I guess that's something we use, right? Topologically.
Yeah, I've always used that as a general term.
Yeah, I do. I like it.
Yeah.
All right. Something in the news which I wanted to bring up. This was forwarded to me at the beginning of the week by Islam Hussein, and it's an article in The Guardian. And by the way, when you go to a Guardian page, they ask you to give some money because they don't charge subscriptions. And I wanted to ask Alan what he thought about that. Is it a good cause to support?
They're a decent newspaper. They're British. They're kind of a British, Los Angeles Times, New York Times type of level of reporting most of the time. I don't know. It's a reasonable cause. I have some larger concerns about the holding out a tin cup business model of supporting journalism. Not really sure that's sustainable. And of course, the problem is that the whole advertiser-supported model has collapsed and people don't want to admit it. And now we're trying to figure out how to pay for news reporting. But this story is certainly timely. And interestingly, I don't get the ping to donate.
No, I do.
Down at the bottom, there's a...
The first time I went, there was a big banner that obscured the content. You had to get rid of it, which...
I like The Guardian, and frankly, their tin cup is tempting for me.
That's not a... If you're gonna give a buck to something, that there are certainly a lot of worse causes you could pick.
But the Times also does something, right? You can see four articles or something, and then you have to...
Right, unless you reset your browser.
Right. Well, they do have ads here on The Guardian too, right? So
sure.
We're making some money. I just don't know where we're going, but I'd like to support some sites, right? And I often come across The Guardian. They seem to have reasonable science coverage, right?
Yeah.
I mean, this article, the title is "Disgraced Anti-Vaxxer Andrew Wakefield Aims to Advance His Agenda in Texas Election." So he is... apparently running for some office. It's not clear to me.
He's not actually running for office the way I read this. Okay. But he is sticking his nose into and one race in particular that's I read through this with great interest because I'm becoming interested in Texas politics, which is its own kind of weird. There's all the links through this, most of them are to other Guardian articles, and they're all very good. But there's one race in particular in Houston, in a particular district, that in the primaries pits a four-time state Republican representative, Sarah Davis, against and she's a moderate Republican, against a much more conservative Susanna Dokupil, Dokupil, and they're focusing in on the issue of vaccination and whether or not there should be. Texas voted in liberal exemptions about, and I think it was about 2003 or something like that, and the vaccine coverage has gone down significantly, even though on average it's higher than the U.S. average. It's has been dropping, and there's Sarah Davis is knowledgeable about vaccines and has fought various attempts by anti-vax people to legislate against vaccination or compulsory vaccination in one fashion or another. And Susanna Dokupil is getting support from this organization called Texans for Vaccine Choice, which just gives me the creeps. At any rate, from what I can tell, Wakefield is just sticking his nose into this, and supporting the more conservative candidate.
Because I saw the headline and read the first line. He hopes to unseat a moderate Republican. I said, he's running for office. But yeah, no. You're right. But this article is repulsive because it kind of goes into what's going on in Texas. And there's this one factoid here. Since the early 2000s, when Wakefield came from the UK, his medical license was taken away from him. And he came, and apparently he lives in Austin. Mm-hmm. So he's Rich's neighbor. The the rate of Texas children exempted from at least one vaccine has shot up by 1,900%. They have a nice map here showing that. So this is horrible. And I mean, people say, well, we're not anti-vax. We just want choice. But it's the same thing, really, because as Hotez says here, and this is very good, he says the side effects are minimal and far less than actually getting infections themselves. So,
one interesting thing is if you look into this Texans for Vaccine Choice, there's no science in it at all. It doesn't talk about vaccine safety or anything like that. Their pitch is that if people cannot opt out of vaccines, then if they don't get their kids vaccinated, people can call them stupid and anti-vax and that kind of stuff, and they don't want to be labeled in that fashion. So they want to be legally stupid. Okay, I mean, from my point of view, if you do, regardless of what the law is, if you do something stupid, somebody's likely to call you stupid. Okay, and this qualifies, but it's nuts. It's just totally nuts.
There's something here about Wakefield getting paid by these autism societies, right?
That looks like where he's getting a lot of his. He's on the board of directors or advisory board of several different autism establishments, and he's making six figures from some of these.
Well,
So
the important thing to understand about him is that he has made his living drumming up a fear about vaccines since the mid-90s.
right.
The original study that catapulted him to fame and caused this round of problems that he forged and put in the Lancet was funded by would-be plaintiffs' attorneys in the UK. There's a fund for this, and this has been well documented by Brian Deere, who's an investigative journalist who did the Freedom of Information Act request and got the data and was just floored by how deep this thing went. That they had financed him for millions of dollars to try to drum up a scientific case against vaccines that could then be used as the basis for lawsuits in the UK. That was the whole scam.
Right.
Ever since then, he has been making his living by promoting this concept.
Believe
And
this is what he does.
it seems to me that he has to know that it's bull. Okay, so I mean, he can't possibly believe in his soul what he's saying.
Well, I don't know about that.
Well,
think about snake oil salesmen. That they have to know that it's not right.
Exactly. Okay, so that's my definition of a crook. Okay, is somebody who knows he's doing something wrong and does it.
I think there's a porous boundary there where somebody starts out running something as a deliberate scam, but after they've been doing it long enough, I think some folks may get to a point where they actually believe their own hype.
Mm-hmm.
And I'm not sure if he's to that point yet. It's impossible to tell because, of course, he can't. He's never going to display any hint that he knows that he's running a scam. But he certainly is, and he's been pulling it off for a couple of decades now.
The other thing that interests me about this is the is that the whole vaccine thing has become political.
Yeah.
Okay.
Yes.
In particular, that it's now anti-vax is now an ultra-conservative issue when it, as you pointed out, Alan, starts off more in the sort of the hippie liberal sector.
Right.
So that's interesting. And to me, there's a lot of these things like that and guns that are, I think, a sort of I get the sense of a sort of a bait and switch thing where if you want to get rid of a moderate Republican, you identify some sort of emotional issue that you can get people interested in and latch onto that and attach it to this individual to try and get rid of them.
Mm-hmm.
So
I like this. At the end of this article, they have some quotes from a mother of 2 boys in Austin, Genie. She's the founder of Immunized Texas, which advocates for tighter vaccine requirements. And she says, I'm part of a lot of the moms networks and groups. It's still incredibly difficult to come out and say vaccines are safe. The biggest challenge we face is if you go on Facebook or Google, you do a search for vaccines, the anti-vax stuff outnumbers pro-vax stuff. So I have an idea. You want to get Genie on TWiV when we come to Austin? 15 minutes,
Sure.
have her come up and tell us what she's doing as a way to publicize.
Yeah, sounds good.
Why not?
Might as well. She's there.
Yeah.
That'd be great. There's another quote in one of these articles that I like, which is, the reality is the bugs don't care about your politics. That's from George Santos, the president of the Harris County Medical Society.
Yep, they are. What's the word for both parties? Vaccines are
apolitical?
No. When you have a bipartisan. Vaccines
bipartisan.
Vaccines are bipartisan. All right. Well, thank you, Islam, for that. Appreciate it. I'll see if I can get in touch with Genie. That'd be cool. We have a snippet for you and a paper today. And this is a real snippet also. It's published in Nature Medicine. And the title is Genital Inflammation Undermines the Effectiveness of Tenofovir Gel in Preventing HIV Acquisition in Women. And this is a multi-center collaboration. First authors are McKinnon and Liebenberg, and the last is Passmore, and they are from the Center for AIDS Program of Research in South Africa, which is in Durban, the University of Manitoba, the University of KwaZulu-Natal, which is in Durban, the CDC, University of North Carolina, Chapel Hill, University of Cape Town, some place called Columbia University in New York, and the National Health Laboratory Service of Cape Town. So HIV, we haven't talked about HIV in a while.
No.
And I've found this really interesting. It all has to do with trying to prevent infection. 35 million AIDS cases globally. Over half of them are women, and a good fraction of those are in sub-Saharan Africa. And it's really hard to get proper prevention to work there. And so we're looking at pre-exposure prophylaxis, or PrEP. And these involve taking a pill before you're at risk, or vaginal and oral interventions like vaginal rings and gels in the case of this paper. And there have been a number of clinical trials to see if they work. In other words, do they prevent infection? And the particular one we're looking at here, 1% tenofovir gel. Tenofovir is a non-nucleoside reverse transcriptase inhibitor.
Actually, it's a nucleoside reverse transcriptase inhibitor.
It's not a non-nucleotide?
No.
I thought it was.
No, if you look it up, it's got a base hanging off there and a bunch of spinach hanging off of that. I'll look it up again.
Look it up, because the one I found said a non-nuke, but I believe you. But look at
I always look for that.
Nucleotide, you're right. It is a nucleotide. I misread. Okay, thank you. So what they do here is, all right, so the different trials of this tenofovir gel have been really wide in their magnitude of protection, from minus 49 to 86%. So no protection to some. And so,
right.
And what does it mean when it's minus 49?
It means that
it's
it
just
appears
promoting
to
it.
Put you at a higher risk. Which, which there's a fair amount of social science data supporting an obvious mechanism for that. That people think they're protected and therefore take bigger risks.
Mm.
Whereas if you don't think you're protected, maybe you don't. But these would be, hopefully, a lot of these would be placebo controlled or something like that, so you could adjust for that. But yeah, there's this massive range in the studies where the gel is either a terrible idea or a great idea, and what's going on.
Mm. So there are a lot of reasons why you would have this range. They talk about them in the beginning, like age of the person, these PrEPs are lowest effectiveness in women less than 25 years of age. The route of exposure,
which of course is the highest risk group,
right? The the amount of adherence. So if you tell people take this gel, apply this gel, not everyone's going to do it, right?
Right.
The particular mucosal target that you're going for, how much penetrance of the drug, and so forth. And I think adherence is a big one that's thrown around. That people didn't do it, so it didn't work. But this paper questions that. They say not really only adherence. Let's look at something else. So here they're looking at inflammation in the female genital tract and seeing if that influenced the efficacy. And they basically didn't redo the trial. They went back to the trial. They have the numbers and they have some samples. They have lavages, basically, which they can take and analyze for pro-inflammatory cytokines as an indicator of inflammation.
And they'd also they had checked for or asked about inflammation at the time of the study, right?
Right.
So they had was there inflammation, and we can look for cytokines.
Yeah, and the cytokines are a good independent judgment of that, right?
Right.
This, by the way, comes from the study is called Caprisa 004.
Yeah.
And
right.
It was a phase 2b double-blind randomized placebo-controlled study with 900 women that took place over a 2-year period from 2007 to 2009. And I forget where. It was in Africa somewhere.
I think it was South Africa.
Yeah.
South Africa. Okay.
I think. I'm not certain of that. I didn't get that from the paper, but
and one of my questions reading it is whether there were several samples during the course of the study of individual women. And the way I read it, it looks like there were, because if you just present at the beginning of the study and you got inflammation, or you've got inflammation, you don't necessarily know whether that's just a transient event or going to carry on.
Mm-hmm.
My sense was that they sampled these subjects several times during the study, and we're talking about a continuing inflammation, I think.
Yeah, I think
that would make sense.
In the limitations part of this paper, they talk about not having multiple samples for everybody. Right. So that was one of the things. If they were going to do it again, which they suggest be done, that you would have many more samples. So they looked at 9 pro-inflammatory cytokines: IL-1 alpha, IL-1 beta, IL-6, TNF, IL-8, chemokine 10, CXCL10, monocyte chemoattractant protein 1, macrophage inflammatory protein 1 alpha, and MIP1 beta. And they say they define inflammation as elevated levels of greater than or equal to 3 of these 9 pro-inflammatory cytokines. And so they have the data for the efficacy of the tenofovir gel, and they can then try and match that up with these cytokine levels using a variety of statistical regression analysis programs. So the question is the gel protective on the basis of whether you have or do not have genital inflammation? And they massage the numbers in a number of ways, in several ways. And no matter how you do it, it looks amazing to me anyway. And they have this lovely, unfortunately you can't get this paper, but they have an amazing graph which shows the efficacy depending on how many cytokines are elevated from 3, 4, 5, 6, 7. You can see, blue is no inflammation, pretty good range from, 0 to 50 or 60 or 70% protection. And the inflammation, the more cytokines that are elevated, the less and less efficacy you see, down to negative, past 200. It's really striking, right?
And good color choices on the graph too. Inflammation should be in red. I mean, little things like this.
Yeah, I totally agree.
To interpret. But the interesting thing to me is that the no inflammation group is a pretty clear, here's where they land on this efficacy. Whereas the inflammation group, you have these huge error bars that stretch way out, which seems like it would kind of it kind of gets to this issue of why have the previous studies shown such a huge range, and it may be this is what's going on.
Mm-hmm.
But I just want to make sure I touch on the point about you can't get this paper. It is paywalled, and given the subject and the interest in this, I think that's the wrong choice by nature.
Mm-hmm.
I mean, this is something that is directly medically relevant in a life and death sense to people, especially in developing countries where they're going to have a hard time accessing it, and they've locked the darn thing behind a paywall. And I think that's totally inappropriate.
Mm-hmm.
Yeah, I agree.
Anyway.
So just a few numbers. The overall efficacy was 34%, but when you stratify according to inflammation, when you have greater than or equal to 3 cytokines, the efficacy was 3%, and the interval, the errors were minus 104 to 54%. And if you have less than 3 cytokines, you have efficacy of 57%, and so forth. So you can see that clearly that's the conclusion here. The best effect of the gel is no inflammation. The other criteria is whether you do it or not, whether the ladies do this or not, who actually used it, because not everyone used it. This is this gel dosing is per coital event, so you have to have it before a sex act, and that doesn't always happen. And they had some numbers.
Actually, the prescription is before and after.
Before and after.
Before and after.
Yeah.
Right.
So it's whether or not they adhere to the gel regimen, not whether or not they adhere to the gel, as they said in the abstract.
And, even among individuals without inflammation, the ones who adhered to the regimen did much better as well.
I mean, that effect is striking, the difference between adherence and not.
Well, yeah. I mean, because when you don't use a medicine, it's usually not very reliable.
Right.
So that is really a very powerful summary.
Actually, right? Yeah, the adherence aspect is is in a sense a control, because it does show that the medicine has an effect.
Right.
It's used.
So a couple of interesting conclusions. First of all, can we fix the PrEP in some way so that inflammation doesn't affect it, right? That would be, because I think it's hard to change inflammation or adherence, but maybe if you made a PrEP that was independent of those, you would do better, although adherence is a tough one to cover. Right?
Well, and it strikes me that inflammation can be part of larger health issues, and maybe what we're really looking at here is the limitations of providing these types of single interventions for a single disease. Instead of focusing on the general context and saying, let's improve overall health care so that not as many people have inflammation when they're exposed to this.
Yeah.
Because that was a very strong predictor of getting HIV.
Yeah. So an interesting question. They don't know what they say. The causes of inflammation remain unclear.
Right.
And one of the things is dysbiosis, an altered microbiome, which could impair the uptake of the drug or something like that. They also mentioned that the viruses that are infecting women with inflammation seem to be less fit than in other cases. So, normally those viruses wouldn't be infecting them. So this is another interesting issue. And they say, maybe we have to look at anti-inflammatories, but I'm not sure that would be a good idea, right? Because that could exacerbate other things as well. So,
right.
But certainly in the future, as they say, if you do another trial, you should stratify the individuals by inflammation to really get an idea of how it works. So I think this is a very powerful study. From my, eyes, inexperienced in terms of epidemiology, and that's why I thought it was important to mention.
Yeah.
All right. Anything else? Okay. Now we have here a paper that has been submitted to eLife, which you can go to the journal and get it. Now, it's the first time I've done that. I don't know how long they've done this. Does anyone know if they have had submitted papers there for a long time?
I don't know.
Yeah, it's interesting. It is.
It's not yet published. It's only submitted.
Yeah, there's a big banner across the top of my PDF that says manuscript submitted to eLife.
Oh, mine doesn't have that.
Really?
Mine's clean.
Either.
So yours is. I just downloaded it yesterday too.
When did I download this? I think I downloaded it today.
eLife Sciences. Let's see. Let me just search and see if it comes up again. Maybe it was accepted in the meantime. Bloom.
Yeah.
And here's the paper. So if I go to PDF, let's see, where's the download link?
There's a little download icon in the upper right corner.
Yeah,
There
I
you
just
go.
typed in the
Article PDF.
PubMed ID, and it still doesn't have any heading.
Article PDF. Oh, so now it doesn't have that heading. Okay, fine.
Okay.
So maybe it's accepted. However, at one point it did have submitted, so it must be up there when it's submitted, which is a new thing for me anyway.
Right.
Extreme Heterogeneity of Influenza Virus Infection in Single Cells by Alastair Russell, Cole Trapnell, and Jesse Bloom. They're at the Fred Hutch Cancer Center and the University of Washington in Seattle. Single virus infected cells. And this paper is interesting for two reasons. First is the tech, which is really pretty neat.
Yes.
Mm-hmm.
And the second is that what they actually find. And so, people are really interested in single cell sequencing, whether it be genomes or transcriptomes, and the technology has been developed to do that. So the question here is, if you infect cells with influenza virus and you look at single cells, what do you see in terms of virus and cell transcripts?
And just as background for the uninitiated, ordinarily, most of the virology that's out there that we look at is done with high multiplicity infections, where you try and infect every cell, so you see the maximum virus stuff in a single cycle, and then you do a bunch of measurements on that population of infected cells. So what you're seeing is an average, and an average done at a high multiplicity infection. And if you think about it, when you infect an animal, that's not at least initially, what's going on. And so it's quite a relevant question as to, okay, what happens in individual cells that are just infected with one or maybe two particles?
Yeah.
Yeah.
So now we have the tech to really do it. Now,
we'd been studying these in bulk because there's too much, you need enough signal to be able to read it. And now that we can actually get a complete genome sequence from a single cell or what have you, we can look at what's really going on in an individual infection.
Yep. So the tech is very interesting. There's a couple of papers that I found, but it's called Massively Parallel Transcriptional Profiling of Single Cells. And this is using a gem code technology. That's the name of it. It's a microfluidics platform where basically, and tell me if I'm getting this wrong, you take your cells and reagents, you pass them through this microfluidics chip, which has multiple channels, and they separate single cells through a very narrow microfluidic, and then each cell gets put into a droplet, which can be collected, and the droplet has all the reagents in it in order to do the sequencing.
Right.
That's the way I understand it, yeah.
Yeah, and you also put in beads, little beads that are festooned with some kind of a marker you can track.
Right.
So then each droplet has one cell, a bead with your marker, and whatever other reagents you're putting in.
Now, in this figure that I found in this paper, they have some droplets without any beads, so I guess you throw those away. Is that right?
I guess so, yeah. Statistically, you may not get a bead in every droplet. You don't want multiple beads in one droplet, so you put them in. It's like doing a viral infection. You put them in at a multiplicity that gives you the outcome you want. And I linked to a page from a company called 10x Genomics that makes the particular system used in this paper. They have about a 3-minute video. The first minute and a half is just hype, and then it starts into the actual, a little bit of nuts and bolts animating how the system works.
So according to the paper, each droplet you have primers with a unique barcode that tags all the RNAs from that droplet, and you're going to do reverse transcription and eventually sequencing. You also have a unique molecular identifier that's put on each mRNA during reverse transcription. And then you sequence just the 3' ends of the mRNA. And since this is an infected cell, you're going to get both viral and cellular mRNAs sequenced.
And once again, for the uninitiated, these barcodes and unique molecular identifiers, what they are is just sequence.
Right.
So you're attaching primers in order to do amplifications. Primers being little bits of DNA with sequence, and you can, for each cell, have a different sequence or barcode that identifies that cell, and for each messenger RNA, a unique barcode. This fascinates me. It's
and
just amazing.
Because you're doing DNA sequences with a fairly short sequence, you can come up with huge diversity of possible sequences or barcodes. And then the output that you get from this is millions and millions of reads, each of which has a short sequence that you put there, plus a little sequence that came from the cell, because you've got sequence information from the, you know what the cells are, you know what the virus is. So you can say, oh, well, okay, that's a transcript of this gene from cell number such and such. And of course, you use computer algorithms to deconvolute all of this.
So the beads, the droplets with the cells are all mixed together back to do the sequencing, right?
It's just what I was going to say. That's my assumption.
It looks
is
like
that once you're done making the libraries, you just mix the whole thing together and sequence it in batch,
right,
and then sort it out according to the barcodes.
Right.
Exactly. Right.
Yeah, because they feed this into just a standard, I think it's an Illumina sequencer. And that's one of the things that they mentioned in the 10x genomics video, is that this slips right into your regular sequencing workflow, but it gives you the ability to track single cells now.
Right. Now, the one thing they did here that's interesting, so they want to know if a cell is infected by one or more virus particles. So they introduced into the viral genome and another barcode at the 3' end of each mRNA, so that it would be sequenced. All right. These are insertions that don't affect viral growth. So then, if you have a singly infected cell, it's going to make mRNAs from either wild type or this or synonymously barcoded virus. And when you get multiple infections, you're going to get both wild type and synonymously barcoded.
And they confirm that their barcoded virus grows just as well as wild type.
They set up all their experiments with an equal mix of the wild type virus,
that's right,
and their synthetically barcoded virus.
Yeah, so if you see both, it's multiply infected, and if you see one or the other, right, then it's singly infected. It's pretty neat to do that.
Now you could have, and they address this later on, there's going to be you will see a certain number of multiply infected cells, but they point out that there's probably, or you see a certain number of cells that are infected with both types of virus. And they point out that there's probably twice that number of multiply infected cells, because some of them are multiply infected with the same virus.
Same virus,
right.
But you can do that. You can figure that out more or less computationally.
The other thing they did was to make sure there were not defective particles in their stocks. Remember a couple of episodes ago, we talked about that, and how if you grow influenza at high multiplicities, you get deletions in the viral RNAs, and that's not good for infectivity. So they did low MOI passage to produce stocks, and they also made sure by a number of ways that they had low amounts of DI particles. All right, so they infect cells at a low MOI, which I had to search hard for, and I found 0.3 HA units per cell. Did I find the right number?
Yeah.
Which I find strange that they didn't use infectivity, because HA, of course, is not a measure of infectivity. It's binding of virus to red blood cells. But at an MOI of 0.3 HA units per cell, if you use the Poisson distribution, 74% of the cells will be uninfected, and 22%, well, they're going to get one HA unit, I think. So I don't know how that translates to being infected.
Yeah, actually, not only is it not infectivity, but an HA unit, it can be quite a few particles, right?
Yeah, that's right.
So you really don't know what the multiplicity is.
Well, I think people have done for their virus. You can do HA versus PFU,
right?
Right, and get a number, but they didn't tell us what that is here. But I don't see if you have PFU, why bother using HA at all? I don't know.
I don't know.
It's funny, but anyway, it's a low MOI, so a lot of cells.
By the way, when they do titer virus in this, they use TCID50 instead of PFU. And I was thinking of, you're so crazy about plaques, Vincent, and you worked on flu. Did you just do plaque assays? Did you ever do TCID50s?
No, I never did TCID50s. I would do an HA once. We would harvest our eggs, do an immediate HA to see if it grew at all. And if it did, then we would do a plaque assay for sure.
Well, with all this avoidance of plaque assays, maybe they're just trolling us.
I had to tell a story. Go ahead.
No, you go ahead.
So Jesse Bloom, the senior author on this paper, gave a job seminar here about 10 years ago. And I remember him in his seminar saying, it was too much work to do the plaque assay, so I developed this other assay, which was some GFP-based assay. So I suspect that is influencing other people in the lab, so they don't want to do plaque assays. I don't know, maybe. I never forgot that he said it's too much work to do a plaque assay.
Did you call that out at the time?
No, I didn't, of course.
Oh,
well,
okay.
It's interesting because Adam Loring does things here by TCID50, and I asked him about that because to me it seems like a little bit more work to do a TCID50 than a plaque assay, but I think it's all just what you're used to. And I can never remember the argument exactly, but he thinks he gets better results with the TCID50 than a plaque assay. I mean, that it's scientifically that there's a little more validity to it.
I mean, it's okay. It's an infectivity assay, so
sure.
But HA is not, so
right.
And once again, for the uninitiated, TCID50, you do a serial dilution and infect cells, and then examine them to see whether or not they look infected, whether there's a cytopathic effect.
Right.
And then there's a cutoff where you're only seeing half of the wells that are infected, and that's a TCID50.
Right.
So, but importantly, it's an infectivity assay. But, dude, I like plaques,
I know we all do. We like it, but, other people do what they want. And all that matters is they get funded and their lab keeps going, right?
Right. And we should say it is a valid way to check a viral titer.
Sure.
And I just found my email from Adam. Number 1, I think they're more precise. Number 2, they don't require as many cells. Number 3, if you're looking at mutants, you can be biased against ones that form small plaques. Downside is that TCID50s take longer.
I don't know. Why would it be more precise than a plaque assay?
Well, he just says he thinks they're more precise.
Okay.
So I don't know.
Well, I don't know. I count the small plaques,
Yeah. Well, if you have good eyes.
Yeah, that's right.
If you have old eyes.
Like us.
really.
Well, yeah, maybe I don't count plaques anymore, so. All right, so the experiment to infect cells at a low MOI, and then with wild type and barcoded, right? And then you collect cells at 6, 8, 10 hours post-infection. You do your sequencing. And here's the story. So I have to say at the outset, speaking of old eyes, I couldn't see a lot of these data because the graphs are too small. And I know you can blow it up, but then I can't read anymore. So I don't know what to do about that. But anyway, I take their word for some of these things. So they look at 3,000 to 4,000 cells per sample. That's the number of cells that they're able to do sequencing for. Most of the cells don't make any viral mRNA, so that you would expect. It's a low MOI. The amount of viral RNA per cell increases with time. You expect that, right? Virus is replicating. And what they say is really interesting is how widely the number of viral RNA molecules varied among infected cells. Less than 0.1% of most cells constitutes viral RNA, but for some cells, it was half of the RNA. So these outliers and you can see on the graph that there are dots up here and there where outliers. But at every graph, I can agree that there is a lot of uninfected cells.
Yeah.
I can see a lot of dots at 0. And then the number of dots that go up increase with time. So it's interesting that there's not a lot of viral RNA in many of the cells.
Right.
So I wanted to know from the very beginning in the introduction when they said from the bulk types of high multiplicity experiments, it's thought that there's 50,000 to 100,000 viral mRNAs per cell. So I wondered, after I'd read the whole paper and went back and reread the abstract and introduction, I thought, what number did they find? And here the range seemed to be as high as 17,000 per cell at the 10-hour time point. So then I wondered, for the bulk number, is that at a 10-hour time point? And what is the time frame of a influenza infection? Is 10 hours, is it done? Or
I think
Vincent, do you remember?
I do think that 10 hours is pretty much done. Yeah.
So these numbers are moderately comparable to what's been found for the bulk thing.
Right. So when we look in bulk, we see this, okay, each cell has this, tens of thousands of RNAs, but then it turns out when they look individually, now that they can, that's a tiny number of cells has a huge proportion of viral mRNA, and a whole lot of other cells have little or none.
Mm-hmm.
And at various times in molecular biology, that kind of phenomenon has been called jackpot. So there's some cells, jackpot cells, that have a lot.
That's right. They
call them in the editorial summary the Bill Gates cells.
Bill Gates cells. That's right. Yeah. Bill Gates cells. They go through a couple of things to make sure that there's no leakage from cell to cell, and to make sure that they're looking at infected cells. I don't want to go through that.
I just wanted to ask, the leakage, is that they think that the RNAs leak out and then are just sticking on the outside of cells? I mean, is that what they...
I suppose either sticking or being taken up, you think?
Yeah, well, that's what I couldn't quite figure out.
Yeah,
so
they just said,
leakage. And I thought, did they mean stickiness?
Yeah,
I guess.
I don't know. But they rule it out, right?
Yeah.
And they make sure that the infected cells were truly infected. And they still, even when they go through this, they still see this extreme variation in the number of transcripts. And here they use what's called a Genie coefficient to quantify the heterogeneity of the distribution.
Yes.
And here they come up with a sentence which I have not seen before in a paper as a fun point of comparison.
Yes.
Has anyone ever seen the word fun in a journal paper?
I've never seen the word fun in a journal paper before. It's great. I love them for putting that there, and I love Eli for running it. So the Genie coefficient, they cite Genie 1921, which is, I didn't look that up, but it's GINI. I assume this is a mathematical or economics paper.
Yeah.
And this is often used to characterize wealth distribution in a society. So you look at distribution of any variable. If everybody had exactly the same amount of money, then you'd have a Gini coefficient of 0. And if one person had all the money and nobody else had any money, you'd have a Gini coefficient of approximating 1. So then coefficients between that are showing you the degree of wealth inequality within a society, or in this case, the degree of infection inequality within a culture.
So everybody should think for a minute what they think the wealth inequality number or the Gini coefficient would be for the United States. And then what you find out if you read the eLife Digest at the beginning of the article, it's 0.47.
Right.
And then the Gini coefficient for the viral genes expressed in different cells, the value is 0.64. Telling you that there's more unevenness in the viral gene expression for flu than there is in income inequality in the U.S.
Great. I like that. Very good.
That is a very fun point of comparison. I like it.
Yeah. So this heterogeneity, they go through a number of possible explanations, and they're kind of technical issues mainly that, and they basically ruled them out. And so then let's talk about the results, what they mean. So this is, of course, a segmented RNA genome. And so, did I not, it's coming later. Yeah. It's a segment genome. So if a segment's not made, of course, if it's not packaged, it won't be able to be expressed in the cell.
Right.
And it's been shown before that most infected cells do not express at least one gene. So they looked at what genes were being expressed in these infections, And so
throughout this, the assumption seems to be that if you're missing expression of a gene, that's because it never got there in the first place. Am I reading this correctly?
I think that it's there, but it's not getting expressed.
Okay.
Yeah, I mean, that's one possible explanation, of course, but I don't think they favor that.
Okay.
I think that it just doesn't get expressed. And in fact, in the end, they call it stochastic something. What is the word?
Yes. Right. The word stochastic appears a whole lot in here.
It's not expressed because
it appears randomly.
Yes, it does.
Stochastically.
It appears stochastically, yeah.
So stochastic is just a fancy word for stuff happens.
Yeah, and we don't know why.
We don't know why.
Right. So they want to get at this randomness. So they look at the amount of RNA for each segment, which they can do. They have the data for that, right? And here's what they find. So there are 4 segments involved in RNA synthesis, right? And they encode proteins that include the polymerase and other things that you need to make RNA. And they say, for example, so this whole RNA plus the 3 or 4 proteins called the RNP, the ribonucleoprotein, and cells that lack any of the genes that make up that RNP, that 4, never have more than a few percent of their mRNAs that are viral. And so the idea there is you need those genes for viral amplification. So if one of them's missing, you're not going to get a lot of transcription. But the other genes, if they're missing, so Rich, sometimes the gene is not seen at all in the transcripts, but I think they think the gene is there, or maybe it's not. I'm not sure.
Could be either way.
Either way, but if you lack one of the other non-RNP genes, you can still make a lot of viral RNA. So the other genes are not so important for high levels of mRNA synthesis. But then they say, Rich, gene absent doesn't explain all the variability, since even cells expressing all genes have a very wide distribution in the amount of RNA that they express.
Right.
Right. And it can range from less than 1% to 50%.
I'm relieved that when you're missing a gene required for RNA synthesis, that you don't make much RNA.
Wow.
That to me is sort of a validation piece of data, you know.
I can't tell if you're being sarcastic.
No, if I were doing the experiments, that's exactly how I would feel.
Yeah, that's true.
Because, you know, the way my life goes, it wouldn't work out that way.
So here's another interesting number. The fraction of infected cells that fail to express a certain gene. And between 5 and 17% fail to express any one of the 4 non-RNP genes. That's outside of the polymerase genes. So it's quite a bit.
Yeah.
Let's see. If we extrapolate this frequency to RNP genes, we would predict that 35 to 50% of infected cells express mRNAs from all 8 genes. So that's not so bad, 35 to 50%. That's in the realm of, you know, defectiveness and problems and so forth, mutations. Okay. The next thing they looked at is the relative amount of RNAs across cells. So even though you have this cell-to-cell variability, the fraction of viral RNA from each gene is consistent across cells. So in other words...
You mean one segment relative to others?
Yeah, right. Exactly.
Yes. Right.
And they actually make there's previously a hierarchy which was published, which is M is the highest, followed by NS, then NP, then neuraminidase, hemagglutinin, then 3 polymerase subunits.
And they replicate that here, which is very reassuring.
Yeah, and that's so that you would expect the replicase subunits to be least needed because they're catalytic, so you can recycle them, right? So that makes perfect sense. Right. And then M is a structural protein, so you need a lot of that. Interesting that non-structural is second, you know, in terms of abundance of mRNA. And then the other cool thing I thought is that sometimes cells get co-infected with wild-type and barcoded virus. Remember, they could tell those cells, and they captured 10 of them.
I really like this part.
And they show all 10 of them in the figure. I love that.
You can, it's great. 10 cells that had been infected with both, and 7 of them expressed all 8 viral genes, and you would have predicted that most would not have expressed all of them. So they say they must be co-infected, right?
Right.
By these, and that must be complementing. You know, one has a defect; the other virus is complementing it. So that's
and
pretty
they show, and this is another nice graph where they show the proportion of each gene mRNA that's coming from each virus.
Mm-hmm.
So, the one that's got half its NA coming from the synonymous and half from the wild type, but all of its M is coming from the wild type.
Yeah. Yep.
It's very cool.
I think this is where I got the impression that they were thinking that if something's not expressed, it's because it never got there. Because they comment that in 4 of these cells, there are shown situations where there was how do I say this? There was expression of one gene from only one virus and another gene from the other virus. And they say that infection could not have happened if there was not a co-infection, because you're showing that the cell had a sole contribution of one of the viruses for each of 2 genes.
So it's a single cell proof or very highly supporting evidence that co-infection can rescue missing viral genes.
Yeah, right. That's their conclusion. Yeah.
That's a relief, isn't it, Rich?
That's a relief as well. I wasn't, I think my relief was over with by the time I got here. I was, and I was into, to be able to look at somehow this particular figure, to be able to look at the fraction of RNA for each gene from 2 different viruses in a single cell, that I like that. That really floats my boat.
It's cool.
It's an amazing level of detail to get out of an experiment.
And think of it this way. So we're looking at 2,000 to 3,000 cells, and maybe a couple of those make a ton of RNA, right? But consider this virus infecting your respiratory tract. It goes in, and maybe a couple cells express a ton of virus, so they have RNA, so they make a ton of virus, and they go on and infect more. So each round, you're going to infect more and more cells. So you're going to have a lot that make a lot of RNA. That's why viruses spread, right? Even though it's rare, it's still in a tissue. There are tons and tons of cells that can be infected and make lots of virus.
I think one of the reasons I like this is because I did a lot of genetics. And so I did a lot of co-infections to do complementation and recombination. And I was constantly, when I would set up one of these experiments, I would have a picture in my head. I would imagine what was going on in individual cells. All right. And here you can actually see it. That's cool.
Right.
The next thing they did, which is quite interesting, they looked at host responses. So these infected cells, of course, they have the sequence of the host mRNAs, and they can see if interferon responses are turned on. And so they look at interferon, and they say, to our surprise, they didn't see a clustering of cells, infected cells, as if the interferon is high in some and not in others.
Right.
And they found only one cell that made detectable interferon.
Yeah, that's amazing.
Right?
Mm-hmm.
They also,
this is the weirdest fact of this whole paper.
Thing. Very strange.
We'll come back to it, but yeah.
And now one of the issues here is that the virus encodes a protein called NS1, which is an interferon antagonist. And so they said, well, could this be playing a role? Is there just a ton of NS1? So they found cells that make a lot of viral mRNA but lack NS transcripts. Okay. And this one interferon positive gene did not have an NS transcript, but the other cells that lacked NS still didn't make interferon. So it's not NS that antagonism that is explaining the absence of interferon. So apparently, individual infected cells don't activate the immune response, even if the interferon antagonist isn't there. Which is
now strange, is that because they're doing a low multiplicity infection? I mean, what's the history of this? If you infect a dish, they say in the paper, if you infect a dish of these cells at a high multiplicity, you get an interferon response, right?
Mm-hmm. You sure do, yeah. Well, if you use lower MOIs, you get far less interferon responses, right? But you are infecting a population. Maybe that makes a difference. as they're talking about autocrine versus paracrine interactions later on. That could be part of it, right? Because in a dish of cells, if you get a few that are highly infected and they make interferon, it's going to diffuse to the other cells and turn it on there. So maybe that's what's happening in a bulk culture and not here.
Yeah.
And they also remind us that they're using these highly purified stocks, and if they had stocks that were prepared at a high multiplicity, they would likely have more DI particles, defective interfering particles that would enhance the innate immune signaling.
Which we learned a couple of weeks ago is important for restricting replication, right? To have lots of DIs, yeah. And so that's something that is clearly important for flu. I wonder for other viruses where you don't have, it's like polio, you don't have much DI particle in the preps. If you did this kind of single cell analysis, if you would see something different in terms of interferon.
Yeah. But so what that means at the organismal level is that the few cells that get infected at low multiplicity at first don't do much with stimulation of interferon, but then the burst from that cell can start to give you a higher multiplicity, and that then leads to the interferon. So it's
sure,
not as early as innate might make you think.
Yeah, we all think those first particles come in and that's it.
Yeah,
that cell makes interferon and they're finished. But maybe it takes more than that. Yep, maybe other things are important early on, like mucus, right?
Or like some of the other host genes they looked at.
Yeah. So
mucus. You say mucus, and I think of Forrest Rower. It's all about mucus.
Yes.
Mucus is important, no question.
It's the duct tape of biology.
I am not disparaging mutinous. Hey, that's good. I like that. The duct tape of biology.
Is it important for adherence?
Yes, it could be. It's actually kind of the combination of duct tape and WD-40. It can make things stick or make them slide.
All right. What about other genes? They found 43 cellular genes that covary with viral mRNA expression, and these are in some interesting groups. Many are associated with the response to reactive oxygen species or hypoxia, management of misfolded proteins, the electron transport chain, stress response. These are all things that are happening in infected cells. So
these all seem like a generalized cell stress type of thing. You've got a whole bunch of viral mRNA and protein synthesis going on. Everything's been cranked up to 11, and this is leading to these misfolding events and this energy crisis and reactive oxygen. And so this seems like it's probably a reaction to that.
And so the interesting thing here is that you can say, not just in a bulk culture, which goes up or down, but here, the more viral RNA you have in a single cell, the more of these particular genes you have. So
and you can look specifically at these outlier cells that have that are hogging all the infection cycle.
That's right. There's one gene they found associated with a cell cycle checkpoint, which is interesting. They don't know what that means. But
this one, it was a little disturbing. There's previous work where people looked for genes that appeared to be involved in flu replication of the cell.
That's right.
Host genes.
None of them were here.
Interestingly, none of the cellular genes that are associated in Watanabe 2010 were identified here.
Those were in bulk culture infections, I presume.
Right. So I think those were bulk culture infections, but it is interesting that nothing out of that shows up in here. Why no overlap? What's going on there?
Yeah. Well, that's going to have to be worked out because maybe there are some fundamental differences.
Yes.
Between bulk and single. So I think more and more people are going to do this.
Both are relevant to the natural infection cycle because, as Kathy pointed out, you've got this. Initially, you're going to have a low MOI infection in your lung. And then after you've got a couple of replication cycles going, you're going to have tons and tons of virus spreading out, and you're going to be doing high MOI infections to adjacent cells. So both of these things are going on in a normal infection.
So go ahead.
I was just going to say, when they looked at these cellular genes, they grouped them by their expression patterns, and they put them in these clusters, but they don't ever discuss what the clusters mean. And then when they talk about the groups of genes, I kind of wish they would have said, they mentioned some gene, NOCIP, and if they would have said, that's in cluster 1, or these cell cycle genes, I think they were also in cluster 1. And it seems like they made this fancy figure with the clusters, but then they never made use of it. And I wondered whether there was more to what their clustering meant.
Right. Okay, so the end is you see this heterogeneity in singly infected cells, and some do not make any viral mRNAs. Some make just a few. Some make all of them. And they say half the infected cells fail to express one or more viral genes. Half of the infected cells, which is really interesting. But why? We don't know why. So they could be missing them. They could have mutations, or, as they say, just inherent stochasticity.
Yes.
Stochasticity.
And this is a viral illustration of the phenomenon that there are a bunch of different ways that something can go wrong, but there's only one way it can go right. And
that's all you need. All you need is one.
That's all you need is for one thing to go wrong. You don't get all these components. For some reason, you're not transcribing all of these components, and you don't get an effective infection. And this is referred to as the Anna Karenina principle. It shows up in a lot of fields of study. It's from based on Leo Tolstoy's opening that happy families are all alike, and every unhappy family is unhappy in its own way. So this notion that you got one thing going wrong, that derails the whole thing. But because you do get all of them happening together, and it all comes together often enough, then in a culture of cells that have thousands of viral infections going on, a few of them are going to carry the virus the next generation.
So are you saying that this happy TWiV family is the same as every other happy family?
Yes, every other happy 2-hour virology podcast is going to be exactly like this.
And I like this sentence. Apparently, stochastic heterogeneity is a common feature of many processes at a single cell level.
Yes.
And that probably goes to neuron firing too, right?
And they cite, 5 papers for that statement, which I thought was impressive.
Yeah. And so then the last thing I want to point out is that interferon doesn't seem to contribute to this heterogeneity, because hardly make one cell made interferon. That's remarkable. Now, why I like this paper, besides really it's great tech, and this is what a lot of people are going to be doing. Having worked on polio, I knew there was a history of people trying to do this in the 1950s. So I went back and I knew this work had been done by Andrei Lvov. And I found the paper in Virology, Volume 1, page 128.
Volume 1 of Virology. Wow.
1955. I was 2 years old. Kinetics of the release of poliomyelitis virus from single cells. This is Andrei Lvov, Renato Dulbecco, Marguerite Vogt, and Marguerite Lvov.
Now, I haven't read this paper, but I'm going to guess they did not use the same microfluidic device.
They didn't.
No, but they made their own Pasteur pipettes from tubing.
Yep.
They used 3 millimeter Pyrex tubing, washed it, sterilized it, and then drew out Pasteur pipettes from it. And then from that, they made even finer points. So maybe they could have bought Pasteur pipettes commercially, but maybe not.
So the way they do it is they have this a cover on a cover slip. They layer oil on top of it, and then within the oil they put droplets of medium containing single cells. And so these are incubated, and the virus is replicating in the cells. And they put the cells there with these fine needles that Kathy just said that they draw out on their own. And so they put single infected cells, and they can watch them. They take pictures of the cells as they break. And in the results section, this is just wonderful. Before considering the problem of virus release from infected cells, let us describe briefly the behavior of normal non-infected cells. Never see that in a paper anymore. Just take it. So they study in this paper, I think, 4 cells. 4 isolated monkey kidney cells infected with poliovirus. We don't know how many polioviruses went into each cell. But the cool thing was they didn't release any virus for the first 5.5 hours. Then within 30 minutes, most of the virus was released. So it was all happening at once. And so and the thing is, there were subsequent papers where they found that some cells don't make any virus. And in fact, they have growth curves for these 4 cells here, and they're very different as well. So it's kind of a first hint that things that happen in individual cells are different. So I just remember this paper and wanted to. And they don't cite it here, but they do cite some phage papers from the 40s, I think, from Max Stelbrook, where he also did single cell infections.
Right. I wanted to comment when we were on the previous paper how recently on TWiV, Vincent, you said you kind of like to look at the last sentence or the last part of the paper, the last sentence. And so the previous paper, they said genital inflammation should be investigated as a potential effect modifier in trials for new PrEP products. Okay, I didn't bring it up at the time, but this paper from 1955, the last sentence, to ascertain more details of the mechanisms of virus release in this virus cell system, many more cells will have to be studied.
I saw that. Yeah, that was great. That was so funny, isn't it? Many more cells. I know.
Yep.
Can you put that in your grant application? We need to infect many more cells.
Many more cells.
More than 4. So that is just great. 1950s. All right. Let's do. Everyone happy with those?
Oh, yeah.
Yep.
All right. Email. We have a email from Kay, who we read one of Kay's email last time. Kay did not like the lizard placenta paper that we did. Dear TWiV team and Nels, I am lagging behind by a few episodes, so chances are that somebody else has already made these comments, which should have been sent in more timely as a follow-up. In the last episode, I listened to TWiV 476 in ACOD 1 We Trust. You discussed an interesting paper wherein the authors claim that multiple viruses upregulate a particular long non-coding RNA, which in turn increases metabolic flux through the TCA cycle. You were totally enthusiastic about the immense workload done by the authors in the elegant experiments. I must say that I'm not totally convinced because this paper dodges a number of important questions. One, the induction of this long non-coding RNA is reported to be interferon independent, which makes sense, but to depend on NF-kappa B. I find it hard to believe that viruses would trigger the NF-kappa B response on purpose to get a metabolic advantage. As a downside, the viruses would have to increase their defense against all the other nasty factors induced by NF-kappa B. Well, that's I don't think that's how it works. The cell is mounting an NF-kappa B mediated defense, right? And that gets rid of a lot of viruses, except the few that could benefit by the increased long non-coding RNA, right?
Right. It's turned an obstacle into a potential advantage.
That kind of stuff happens all the time.
Yes.
2, the authors don't even discuss the fact that their link RNA is sitting next to ACOD1, which has 2 important properties. First, ACOD1, the protein, is also known as IRG1, immunoresponsive gene, and is a well-known NF-kappa B target and regulator of the inflammatory response. Second, ACOD1 acts on cis-aconitate, which is an important intermediate in the TCA cycle. ACOD1 generates itaconic acid, which inhibits the glyoxylate shunt. I don't want to go into details. Here is a good reference. I don't know why that's a problem. Does anyone understand?
I don't know where that's going.
It's just
oh, they didn't discuss this. Okay.
They didn't discuss it.
And that's interesting because it's more metabolism. Okay.
Right. So it could be relevant.
Yeah.
It's worth thinking about.
3, the neighbor relationship of the link RNA and the ACOD1 gene raises a number of suspicions. Does the knockout of the link RNA really not affect the protein coding ACOD1 gene? Wouldn't it be obvious to at least check if link RNA acts by regulating the expression of the neighboring ACOD gene? It might be a total coincidence that both the link RNA and the neighboring ACOD gene or NF-kappa B targets are anti-inflammatory and regulate the TCA cycle, but not even discuss this? I don't know. It's a good point.
That's a good point.
Oh, on a more positive note, here's an answer to a question raised by Rich Condit. Are there other proteins, enzymes that bind RNA? Yes, there are. One of them is particularly pertinent. The cytosolic form of aconitase, the enzyme that isomerizes citrate in the TCA cycle, moonlights as a specific RNA binding protein recognizing iron response elements in the mRNA. In this function, this protein is referred to as RP1, gives a link to that, but is active as an aconitase nevertheless. And it has the same substrate as ACOD1. A coincidence? Maybe not, right? Interesting. Okay, thank you, Kay.
Keep us honest here.
That's right.
Keep listening.
Alan.
Justin writes, just wanted to say I absolutely loved the section of TWiV 480 where Scott Hensley was talking about the RBC assay and other tests they do on influenza. That's exactly why I listen. As a total nerd who does method development in a pharmaceutical lab, thanks. Hope to hear him back on the episode.
You should like today's paper.
Yes.
It's some cool tech. Kathy.
Bryn writes, hello. My question is this. Although different species of viruses probably had very different origins, could one of those possible origins be that viruses have come from cells as defense mechanisms? I'm watching all of TWiV from the beginning and haven't seen every episode yet, so sorry if this has already been covered. I was wondering if it's plausible that some single-celled organism created a kind of chemical with the, quote, intention of warding off being eaten or destroying nearby cells, and this, quote, weapon's potency lied in its ability to behave as a virus. Maybe the cell was attacking another cell with a very similar genetic sequence, but was just different enough to avoid also being infected. Or maybe built into an animal's genetic code to discourage cannibalism or inbreeding, sort of like a prion. Also, perhaps the cell didn't create a virus for the purpose of defense, but as a means of reproduction that was so effective that in itself became reproducing. As a side note, do you think it's possible for a poison to behave like a virus, somehow being able to create more of itself using an element of our biology and infect cells, but with zero genetic code and be as simple as a chemical compound? I'm planning on going to school to become a medical lab technician, and I'm fascinated with virology, and I would like to keep working my way up through school to become a virologist. I'm incredibly fascinated by viruses, and I think the key to learning about how life formed, to begin with, lies with them. I'm learning about them as a hobby, but currently don't have an education above a high school level. Thank you so much for reading. So, wow, Bryn's in high school, or has all
a
these
high school
profound
education
questions.
is coming up with these very, very interesting questions.
Yeah.
Viruses evolving as defense mechanisms. Sure.
Yeah. Why not? Why not? Definitely.
I mean, we certainly have examples of viruses that have been co-opted for an organism to benefit. Maybe they started out that way. Some of them.
Yeah. I mean, one of the ideas is that they came out of cells, right? And so it could be as a defense mechanism. Why not? Yeah. I mean, you can imagine that because now the virus turns on the cell eventually, that it was originally designed to turn on something else and it got away. But who knows? There's no intent, of course. It all happens with selection. I like that Bryn is watching all the TWiVs. That's great. Poison. What is this? Is it possible?
A chemical that could replicate itself.
So do any poisons?
Closest I can think of is prions.
Yeah.
Prions or some kind of ribozyme type of thing, a self-replicating RNA. You'd have to talk to a chemist about this, but I guess it's possible one could come up with a chemical structure that is not technically a genetic code, but that behaves like one.
Well, it would have to be, if it works like a prion, it would have to mimic something in us, right? And convert
it could either change the shape of something around it, or it could somehow catalyze a reaction from available materials that forms new copies of itself.
Yeah. These are all great ideas.
They are.
If you keep thinking like that, you can be a virologist.
Yeah.
Why not? Very good. All right, last one, Rich.
Fernando writes, Dear Vincent and the Virals. Couldn't resist thinking of TWiV Crew as a pioneering rock and roll band.
Bet.
This bioinformatics paper just popped up in my Twitter feed. It shows some curious biases on how differentiated gene expression is reported in abstracts relative to the underlying expression data. It's not virology, but I wonder whether the same phenomenon plays a role in bioinformatics-based studies you have reported recently. Best wishes to the band. And he gives a link here that I have, I confess, not looked at.
So this is a paper called, it's in BMC Medical Genomics, Differential Gene Expression in Disease, a comparison between high-throughput studies in the literature. So basically, this is a meta-analysis, sort of they mined gene-expressing analyses, and then, which you can, the data, the raw data, and then they looked at the text that describes it.
Mm.
And they conclude that the literature shows a preference for reporting genes associated to higher fold changes rather than genes that are simply differentially expressed. All right, so they're saying, you have this data set, but you're only looking at the genes that have high fold differences.
Right.
And then finally, they prefer genes that are overexpressed rather than underexpressed, that are overexpressed in multiple diseases, and that are in general popular in the biomedical literature. So if you're an unpopular gene, nobody talks about it. Yeah. So that's the thing. And they wonder, so Fernando said, who knows how far this is going. And so it's kind of interesting to think about that. And this is a general issue with big data now, right? You generate all these data and you have to analyze it and you present your conclusions, and few people are going to duplicate that analysis, right?
Mm-hmm.
Yeah.
And so if you want to just look at really highly overexpressed genes, that's what's going to happen. So I agree that it probably plays a role in lots of studies. I don't know how many papers they looked at here, but it's interesting.
Yeah.
All right. Let's do some picks. Alan, what do you have?
I have the Next Generation Science Standards. This has been sitting in my queue for a little while. And I had put in a different link to one of their subpages, which takes you to a video that appears to be broken, at least on Kathy's machine and mine. So I've now replaced that link with their homepage. The Next Generation Science Standards, this is an initiative from the National Science Teachers Association, which is a nonprofit, I guess, that focuses on this. And their concept is that we need better science education, and the way to do that is to have a national curriculum. Of course, here in the U.S., we are blessed with a system called federalism, where instead of one national system, we have 50 individual states, and at the school system, this is balkanized even further down to counties and sub-districts within counties. So everybody comes up with their own system. But this site describes things that are being done to put together more of a unified curriculum, what should be in it, how it should be approached, ways to teach science within a much more modern context than what is often done these days, and updates from different school systems that have either implemented this or implemented something very similar to it. So if you have a kid in school, or you're a teacher, or you have any interest in our educational system because it is fundamental for our future, then might want to take a look at this and see where your state stands and agitate for change if necessary.
Cool.
So University of Michigan was involved with this, Kathy.
I didn't see that.
Michigan at the end, Michigan State and University of Michigan. Yeah. Cool. So Alan, it sounds to me like you think we could never have a federal education standard for science?
Not exactly as such. It is always going to be piecemeal because so much authority is vested in individual school districts, and because we rather perversely handle the funding for our school system at an entirely local level or a largely local level. So that's why poorer communities with less property tax revenue end up with poorer schools, and you cause all these problems. But that's one of many layers of issues in addressing this. What this is doing is saying, okay, let's have a model of a curriculum that individual districts can adopt and promote it, and hopefully get individual districts to adopt it. And they seem to have made some progress. Unsurprisingly, Massachusetts is doing pretty well on this. They haven't adopted this exact set of standards, but they're functionally duplicating a lot of the same principles.
So now that we have a different link, over on the left-hand side of that page, there's View the NGSS, the standards.
Right.
And when you click on that, it gives you this whole grid of really cool stuff. And over on the right side, a whole bunch of things for first-time visitors. And what I like is that they have it broken down by elementary school, middle school, and high school. And at each of these levels, they have something called engineering design. And I have to tell you that I got all the way through high school. And, then I got to college, and there were these people who were engineering majors. And I had, uncles who were engineers, uncle and grandfather and older brother and all that. But, I don't know if it was because I was a girl or I was oblivious or whatever. But, if you had asked me what's engineering, I couldn't have told you.
Yeah.
I'm your control, Kathy. I'm in exactly the same space.
Me too. And I came up in a subsequent generation and had a very similar experience. My family is loaded with scientists and at least a couple of engineers. And I got all the way to college and, engineering sounds cool, but what the heck do they do? So, I'm very glad to see that that's finally being put into a curriculum and my daughter experiences this at school too. They do problems where, okay, here's some physical problem to solve. How are you going to do it?
Yeah.
Another issue, and there are many, but so my two sons got interested in computer science. Everyone knows about computers because everyone's life is surrounded with, or many people, but one course in high school for computer science. And that's not good. Because a lot of kids would take computer science courses and be better prepared when they go to college and want to major in it. I think we're missing out by not having more curriculum in that field as well, and many others, of course. But that just popped into my head. Yeah, I didn't know what an engineer was when I graduated college. What do they do, actually?
I guess that's why none of us became engineers.
Right.
Rich Condit, what do you have?
I picked essentially a fact-checking site. I'm astonished that we haven't picked this before. Many people may be familiar with this. Snopes, which is my go-to site if I want to know whether something is legit. They, for any given issue that you find floating around on the internet or in the newspaper, if you want to know what the facts are or whether this is fake news or not, you go to Snopes.com and do a search, and they are likely to have a story on it. And I think they're pretty accurate, and their research is quite thorough, and they're very interesting. So I use them all the time.
Now, I don't know this. I've heard of it, but how do we know that they're right?
Well, that's the question. And even before I picked it, I snoped Snopes.
Well, that can get very meta, can it?
No, I sort of poked around at articles on different fact-checking sites to get some sort of outside opinion on how accurate these guys were. My understanding is that they're pretty accurate. Yeah. I went. One of the reasons I chose it now is that I came up with two things recently. One was, have you seen the articles about this? There's a scientist from CDC that's gone missing.
Yeah, I saw that. Yeah.
Right. So I saw somewhere, not a brilliant source, not a very viable source, but I saw a thing that said that this guy had, during his history, made an association between the flu vaccine, the most recent flu vaccine, and flu deaths, as if the new vaccine was killing people. Okay,
right,
and now he's gone missing. Right. Okay, so there's this implied conspiracy theory, where the vaccine manufacturers have killed this guy, right, because he's trashing. Because he's saying that vaccines killed people. Well, if you go to Snopes, they have this report on there, and they trace the whole thing down. They said that somebody made up this story some time ago about a CDC scientist who they did not name, who was bashing the vaccine in this way, and now this guy goes missing. So they can now elaborate on that story and put a name to it. Okay, and it's all baloney. And it's unfortunate this guy's missing.
Should be like a built-in sidebar permanently on every Facebook
Oh,
page.
for sure,
Exactly.
Facebook.
Mhm.
So the other story I came up with, sorry, but I got to do this, was a story that started off with why did Mr. Rogers have long sleeves on his sweaters all the time? And the rap was that it was to cover up a bunch of tattoos because he was a decorated combat veteran. And it has all this stuff about how Captain Kangaroo was the same. And this story came from Lee Marvin, who was wounded in Iwo Jima. And yes, Lee Marvin was a World War II combat veteran who was wounded in Saipan, which was just as bad as Iwo Jima. But Captain Kangaroo did not fight with him and was in the military and saw no action at all. And Mr. Rogers was never in the military. You get all that from Snopes.
So I just found this one article on the front. Did Pfizer's vice president blow the whistle on Gardasil? A disreputable website falsely claimed that the vice president of Pfizer said a vaccine for human papillomavirus is deadly. And so this is supposedly this vice president was trashing Gardasil. And one of the lines in the article says, of course, it's unclear how an executive would blow the whistle on a product his company doesn't make. Gardasil is manufactured by Merck, not Pfizer.
Yes.
They're really very good. I agree with you, Alan. It ought to be some sort of app that goes along with your news feed.
Yeah.
Claim, did Ben Carson buy a 31,000 dining set?
Yep.
The answer is yes, and he canceled it, right? Oh, this is great. I have to put this on my RSS feed, definitely. This is so important because this whole business is an issue, right?
Yep.
And as you've already discovered, it's well written and it's entertaining.
Mm-hmm.
Kathy, what do you have?
I chose something. It's a Vimeo video called Dance Dance by Thomas Blanchard. And it was evidently for Valentine's Day, but I'm a little late. Anyway, it shows flowers representing 4 different seasons. So it starts out with fall and a rose on fire. And then probably the coolest, well, all the sequences are cool. Ice for the winter. So it shows flowers being encased in ice. And then some blooming flowers for spring, also as time lapses. And then for summer, they have these color inks falling down onto the flowers. And so it's kind of for Johnny, and too bad Dixon's not here because he'll never know about this one. But anyway, they're really nice, and there's nice soothing music accompanying it. So I just thought it was fun to look at.
It's very cool. And you can find all of Thomas Blanchard's other videos here. And what is really a more civilized site than YouTube. It's really nicely organized.
Yes.
Mm-hmm.
It's clean and easy to find things. And unfortunately, most people are over on YouTube. So that's the way it goes. Did you want to have a reminder now or later?
Oh, I'll thank you, because I'm off that page. And I just wanted to mention that on Vincent's virology blog this week, he has a guest blogger, and that's Trudy. So Trudy had been on TWiV, and it's an interesting blog post about influenza and its ocular forms. And I knew nothing about that before. And so it was an interesting blog post to read. So check it out.
Yeah, she did good job. Very nice. My pick is an episode of TWiV this week in evolution that we did, Nels and I did last week with Sarah Tishkoff, who's at the University of Pennsylvania. And she's in genetics and biology departments, but did a fascinating study published in Science on the genetic basis of skin pigmentation in Africa. And, you know, she said, you have to listen to it, but bottom line is they went to a few different countries. They measure skin color. They have this color meter that they aim at the skin, and they can measure the skin color. And then they take a little blood and they look at single nucleotide polymorphisms and try and identify genes that are involved in different pigmentations. And she said there's a big range of skin pigmentation in Africa. And they found a couple of genes that were really interesting and involved. One of them was a melanin-related gene, but there are others involved in UV resistance and so forth. And it's really interesting how you can measure almost anything in people And look at the genes involved now. It's just great.
Cool.
And she's got this great background. She started out as an anthropology major and then slowly got into, genetics. And you can see how this comes out of it. She's really interested in the different populations and so forth. And she's now going to look at height and weight and all kinds of, hairiness. She talked about. I think this is so cool. We can look at ourselves this way. So, and she's really good. So check it out.
Neat.
All right. That's TWiV number 483. You can find it at Apple Podcasts, microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And if you want to help us out financially, microbe.tv/contribute. Kathy Spindler is at the University of Michigan, which is in Ann Arbor, Michigan. Thank you, Kathy.
Thank you. This is a lot of fun.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. He's currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Would you recognize Wakefield if you ran into him in a supermarket?
No, but I have been tempted to go sort of sniff around his habitat. I think he's got an office somewhere.
Really?
And
Wow.
and find out what's going on.
Alan Dove is at turbidplaque.com. You can find him on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to the ASM for supporting TWiV and Ronald Jenkins for his music, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.